तेजस्विनावधीतमस्तु मा विद्विषावहै। ॐ शान्ति शान्ति शान्तिः।
In the last class, we were discussing about the secondary wastewater treatment systems. The secondary wastewater treatment systems are very important, because they try to remove the organic matter present in colloidal form as well as in the dissolved form. These organic matter which are removed in the secondary treatment system are biodegradable, and hence the microorganisms are one of the reactants for removing the organic matter. So, other reactant that is required in this system is the dissolved oxygen. So, it is essential for us to know about some aspects of microbiology connected with the wastewater treatment system, that is secondary treatment of wastewater. So, in this particular instance, we were discussing about the bacterial growth curve, and we also discussed about, to some extent, kinetics of bacterial growth. So, these are two important things which we should study to understand the secondary wastewater treatment systems which employ the microorganisms. So, in the kinetics of bacterial growth, we did derive an equation, fundamental equation for the growth of microorganisms. Dx by dt is equal to mu into x. This is the equation we developed. So, x is the biomass, weight of biomass in milligrams per liter, and mu is the specific growth rate of microorganisms. The specific growth rate is dependent upon type of food available for the microorganisms and also type of microorganisms themselves. So, it is a function of the food and as well as the microorganisms. Now, this specific growth rate is not a constant, but it is a function of the substrate concentration. S is the substrate, or this is the food for the microorganisms. The food for the microorganisms provides, the food should provide carbon source and energy source. That is what we discussed in the last class. This also could be the biochemical oxygen demand, BOD, of the wastewater is a substrate. So, this is a function of, that is a mu, specific growth rate is function of the substrate concentration, which is given by the equation mu max, mu is equal to mu max multiplied by S over Ks plus S. In the last class, we had defined all the terms. Still, I would like to redefine these terms. This equation is Monod's equation, developed from Michaelis-Menten's equation. In this, mu max is the maximum specific growth rate constant, maximum specific growth rate constant. This is a constant, this is a maximum growth rate constant, and S is the substrate concentration and Ks is the saturation substrate concentration. saturation, substrate, concentration. So, if I were to plot this particular graph, the graph looks like this. That is, if I make a plot between S, that is a substrate, in milligrams per liter. Again, the substrate is measured in milligrams per liter, as well as the microorganisms are measured in milligrams per liter, versus the mu, that is specific growth rate of the microorganisms. Then as substrate concentration is 0, the mu is also 0, there is no growth rate. And as you increase substrate concentration, the concentration of as you increase the concentration, mu also increases. This particular thing reaches a maximum value, and this is called a mu max. So, mu will reach a maximum value, okay, for a specific concentration of the substrate, and even if you increase the concentration of the substrate, the mu will be only mu max. That is, when mu reaches mu max, it is independent of substrate concentration. Till such time, the mu, that is the specific growth rate, is a function of the concentration of the substrate. When it reaches mu max, it is no longer a function of concentration of substrate. This particular equation, this particular curve is nothing but rectangular hyperbola. Equation of rectangular hyperbola. And now we define the Ks here. Saturation constant is nothing but concentration of the substrate, this is Ks, when mu is equal to half of mu max. That is what we had discussed in the last class. So, now we will move forward from this equation. This is what is the equation what we have. This is dx by dt now can be written as mu max S over Ks plus S multiplied by x. So, this is the equation substituting for the mu. So, this is the value of the mu, that is a specific growth rate. In terms of the maximum specific growth rate constant, saturation concentration of the substrate, and the substrate concentration itself, that is what is the equation. Now, here we can consider from this particular thing, this is our fundamental equation, maybe you can call it number 1. Now, we consider two cases. Case number 1, in the case number 1, what I consider is I have unlimited concentration of substrate. So, S is unlimited, that is I have very high concentration of substrate available to me, available to the microorganisms. So, that means plenty of food is available. So, when the plenty of food available, this particular condition is possible only during the initial periods of the reaction, when we have got plenty of food. Now, during that particular time, we can assume substrate is unlimited, as a result of which Ks is very less compared to S, that is in this denominator Ks is very less compared to S, and hence we can say Ks plus S is approximately equal to S itself. So, substituting this in this equation, dx by dt I have now equals mu max over mu max into x. So, this is one equation I have, this is a second equation, I can call it, substituting this Ks plus is equal to S, and then that S and this S cancels out and multiplied by x. This is a first order equation for the growth of microorganisms. This is a first order equation, and this equation can be solved now. I can solve this particular equation because mu max is constant and dx by dt depends upon the concentration of the microorganisms. So that means if you try to solve that equation now, it is very simple. Dx by dt, dx by x is equal to mu max into dt. Then integrating this particular thing at time t equal to 0, we assume that the microbial concentration is x naught. At time t equal to t, the concentration of microorganisms is x. So then solving this particular equation, I have ln x over x naught and with the boundary conditions mu max into t, or x is equal to x naught e to the power of mu max into t. So this particular equation is a first order equation. It represents exponential growth phase of the microorganisms, exponential growth phase of microbes in microbial growth curve. I considered the microbial growth curve last time, that is what exponential growth curve. Again, maybe I can plot this exponential or this growth curve of microorganisms. So that is what redrawing that growth curve and identifying where exactly this equation is holds good. That is t is the time, and this is the microorganism concentration. I have a what is called a lag phase. This is x naught is the initial organisms, lag phase, and then log phase. This is a so not lag phase, lagging behind, and this is a log growth phase that is growing exponentially, and we have what is called a stationary phase and a decline phase. These are the four phases which we have discussed. Just I want to reiterate that particular thing. So this is the equation. Whatever I have written this solution or the equation is this for this particular phase, log growth phase. So this represents a log growth phase. In log growth phase, I have substrate unlimited. Okay, that means this particular curve can be represented by the equation dx by dt equals mu max into x. Okay, this is a log growth phase. Now what is happening afterwards, log growth phase? The microorganisms are growing in log exponential order, and then the food that is available is getting depleted, as a result of which with the time the food gets depleted. So as the food is getting reduced, the quantum of food is getting reduced, we can consider as a second case, case number 2. Case number 2 is when the food is limited. When the food is limited, S is limited. The first case unlimited food, second case limited food. In this, what would happen here in this particular case is the concentration of substrate is less than Ks. That means the concentration of food is less than Ks. Ks is the saturation concentration of the substrate, and the food available is less than that particular thing. In most cases, in wastewater treatment plants, except particularly in the domestic wastewater treatment, we encounter this particular scenario. This scenario is there. While in the industrial wastewater, where the BOD of the wastewater is very, very high, we may get the first exponential growth phase. Here we get the food is limited, food is getting exhausted and it is limited. So when this is the scenario, what we do is Ks plus S is equivalent, approximately equal to Ks itself. substituting this particular thing, substitute in equation 1, I will substitute in this particular equation. What would happen is dx by dt now would be equal to mu max over Ks, mu max over Ks multiplied by S into X. This is what is the equation available with me now. So, K in the denominator Ks plus S is made equal to Ks only because S is very small, and then the numerator I cannot neglect that S. So, I am writing S into X is equal to is dx by dt is equal to mu max by over Ks multiplied by S multiplied by X. So, you can see here there are two variables, one is the S and another is X, and I cannot definitely solve this particular equation like I could solve the other equation. So, mu max is constant, Ks is constant, these two things are constants, this particular value is a constant. However, dx by dt varies, is a function of the concentration of substrate and concentration of microorganisms, because of the simple fact that the food is limited. When the food is limited, the growth rate is a function of the concentration of the substrate. When the food was not limited, then it was not the function of the concentration of the substrate. That is what you can see here very clearly. So, when lot of food is there, then the growth rate is not dependent on the concentration of the food. When it is limited, then it is function of the concentration of the food. Now, S and X, if you try to look at them, what is this S and what is this X? They are dependent variables. They are dependent variables. What do you mean by that? That means as the substrate concentration is increasing, when the microorganisms utilize the substrate and hence substrate concentration is decreasing, then the microbial concentration increase. So, in other words, as S is decreasing, that is what I will show the arrow mark, decreasing arrow mark, then X will increase. Okay. There is an increase in the concentration of X because of decrease in the concentration of S, that is why they are dependent variables, and hence I can derive an expression between them, between S and X. So, that expression is nothing but what is called yield coefficient. Yield coefficient. Yield coefficient gives a relationship between S and X. Yield coefficient is given by weight of biomass produced, biomass is X here, I will put it X, produced per unit time, in a given time, divided by the weight of, I will put it here, weight of substrate, that is the food utilized by the microorganisms in unit time. So, this is what is called yield coefficient. So, mathematically I can write this y is the yield coefficient, that is nothing but how many kilograms of microorganisms are produced in a unit time per kilogram utilization of substrate by the microorganisms. If microorganisms utilize 1 kilogram of substrate, how many kilograms of biomass is produced? That is nothing but yield coefficient. It is just like crop of the yield of the crop. Okay, this can be mathematically written as biomass increase dx and decrease in the substrate concentration minus ds by dt, and per unit time dx by dt I will put it here, dx by dt I will put it here. So, in other words, y is nothing but dx by dt over minus ds by dt minus ds by dt. Minus sign I am putting, why? Because substrate is being utilized and hence there is a decrease in the substrate concentration. So now this particular equation can also be written as in the finite terms of time. Suppose if I take a finite time, suppose x is the concentration of microorganisms at any time minus x naught, that is initial time, is x naught, divided by the concentration of substrate, again utilization of concentration of substrate, use of substrate concentration, initial substrate concentration is S naught, that at time t equal to 0 minus S. So this is the expression what we have. So in this particular thing, x naught is the concentration of microorganisms at time t equal to 0, x is the concentration of microorganisms at any time. Similarly, S naught is the concentration of substrate at time t equal to 0, and S is the concentration of substrate at any given time. So this S naught minus S is the substrate that is being utilized by the microorganisms. When they utilize the microorganisms, this much amount of biomass is produced. So this is the equation what we have. Let us see little more about this yield coefficient. So when we consider the yield coefficient, the following thing will, suppose if I give the substrate, again I will reiterate, substrate is the food, substrate is a BOD, biochemical oxygen demand, that is present in the wastewater. So when the substrate is given to the microorganisms, that means I will give the substrate to the aerobic microorganisms. Aerobic microorganisms are the organisms which utilize the organic matter in the presence of oxygen, that is why they are called aerobic. When they use this, they will use in this fashion, they will use 50% of around 50% of the substrate for production of energy production. And energy production is manifested by the production of carbon dioxide and water. In any reaction, in the biological reactions, if carbon dioxide and water are produced, it means that the energy is produced. The cell is deriving the energy from the substrate. Remaining 50% of the substrate is going for the biosynthesis. This is biosynthesis. Biosynthesis means increase in this results in increase in biomass. So that is nothing but x minus x naught, increase in biomass concentration, x minus x naught, that is what I have written there in a unit time. That is what is going to happen. In a aerobic system, 50% goes for energy, 50% goes for biosynthesis. If that is the case, can you tell me what would be the value of y in this particular case? The value of y in this particular case is 0.5, that is the maximum value. That means you can see here the denominator is the weight of biomass produced over unit weight of substrate utilized. Unit weight of substrate utilized is this, 50% goes for the energy, 50% goes for the synthesis, and hence whatever is synthesis is the y. Y is a maximum of 0.5. In fact, in actual case it will be less than 0.5, okay, less than 0.5. This is a maximum value that is possible is 0.5. That is the importance of this yield coefficient in the aerobic systems. Suppose if I were to have same substrate, if I give to anaerobic systems, anaerobic microorganisms, the anaerobic microorganisms do not utilize the oxygen. Oxygen is not supplied to them, no oxygen. In the absence of oxygen, they work. When these work in the absence of oxygen, what would happen is they would tend to use 70 to 80% of the substrate for production of energy. For energy production, they use about 70 to 80% of the substrate. An end product of this particular thing could be carbon dioxide, water, and also another gas called methane. Okay. Some energy is locked up in the methane, that is why more substrate is used for the energy production. So, anaerobic organisms are not very efficient in oxidizing the organic matter for the production of energy, while in a remaining part, that is 20 to 30% of the substrate goes for the biosynthesis in the anaerobic system, only 20 to 30%. Now you can see that what is the yield coefficient for anaerobic system? Yield coefficient for anaerobic system is approximately 0.2 to 0.3, even less than that. This is a maximum value. This is a maximum possible value, but actual practice this will be less than 0.2 also. So you can see here the yield of microorganisms in anaerobic system for the same amount of substrate is less, for aerobic system it is more. That means if you are employing aerobic systems for the treatment of wastewater, then you will produce more microorganisms in the aerobic systems and you will produce less microorganisms in the anaerobic system. Okay. This is one of the differences we can right away say between the aerobic systems and anaerobic system. Well, continuing our discussion with respect to the yield coefficient, you can see here from that equation dx by dt or dx by dt is equal to y times minus y times ds by dt. Y times ds by dt. Y is less than 1. Okay. That is why dx by dt is not equal to dx by dt is not equal to ds by dt, but it is less than ds by dt because y is less than 1. Now this equation also written sometimes as y, instead of this negative sign, what I will do is this negative sign I can take it inside and write it as ds by dt utilized. ds by dt u is rate of consumption of substrate or utilization of substrate, rate of consumption of substrate. So when I write u utilize, then I take off the minus sign, that is all. Now these two equations are dx by dt is equal to y times ds by dt utilized. These equations are mirror images. Okay, they are mirror images. Suppose if I were to plot this equation, I can plot it like this, like following. I have what is called this is the time and this is S and this is S also. This axis can be representing both S, microbial concentration and substrate concentration. So both are expressed in terms of milligrams per liter, both of them. This also is expressed, this also is expressed in terms of milligrams per liter. At time t equal to 0, what would be the concentration of S? Concentration of S would be S naught. Let us take this, a maximum concentration. S naught is the concentration of the substrate. As a function of time, with respect to this equation, that means again this is with respect to this equation, that is dx by dt, again I will write it here, dx by dt here, I will write it here, dx by dt is equal to minus y time ds by dt utilized. This also dx by dt is equal to mu max s over k s plus s into x. So this, all this is the equation, dx by dt is equal to this, or minus y into ds by dt is equal to this. So we have this. So that means that at time t equal to 0, substrate concentration is S. If you solve this equation, I try to solve this equation, and the solution of this equation results in this particular graph like this. So this is the variation of concentration of the substrate with respect to time. How exactly the concentration of substrate is varying with time. So, this is for S, this curve is for S. Now, I can also use this equation to find out what is the variation of x with respect to time. So, at time t equal to 0, the concentration of x is equal to x naught. So, this is x naught to start with, and as a function of time what is happening is the microorganisms are utilizing the food and they increase in number. So, that particular number increase in number can be written like this. So, this is the equation for x and this is the equation for S. You can see the difference between these two things, difference is not attaining the same level, x is not attaining the same level of S naught. The reason for that is because of the yield coefficient, yield constant. So, entire substrate is not going into the formation of biomass. So, this is because of the yield coefficient difference in yield coefficient, this is what is happening. So, this is the curve I can represent this particular equation. So, there are mirror images, that is dx by dt growth rate and then substrate utilization rate are mirror images. Now, let us consider one more factor, that is we have said that is y is equal to x minus x naught over S naught minus S. This is what we have said. Yield coefficient is the weight of biomass produced per weight of a substrate utilized in a given amount of time, same time what is going to happen. Now, this particular thing is y into S naught minus S is equal to x minus x naught, I can write it. Now, let me look at a condition when entire substrate is utilized, entire S is used by microorganisms. That means microorganisms have consumed entire food has been completely consumed. So, in that case what would happen, what would be the value of S? S will be equal to 0. S is completely consumed, that means S is 0. When S is 0, okay, when the entire food has been consumed, then what is the concentration of microorganism produced at the time? That x will take a value of x max. That means maximum concentration of microorganisms are produced when the entire food is utilized by the microorganisms. So, in this particular equation, if you substitute x max is equal to x max is equal to this, I will take it as this side, x naught plus S is 0, y times S naught. So, the maximum concentration of microorganisms are produced by as per this equation, okay, entire substrate has been used. So, this is the equation what we can suggest for this particular thing for the maximum growth of microorganisms. So, now let me consider, I have been considering till now the bacterial growth rate, that is dx by dt. Now, I would like to consider another aspect, that is substrate utilization rate. Substrate utilization rate. Okay, substrate utilization rate. Substrate utilization rate according to the equation ds by dt utilized is equal to, I can write it as here as 1 by y into mu max S over Ks plus S into x. This equation I can write it here. So, this is substrate utilization rate is equal to this mu max S over Ks plus S plus x. So, this is one equation I can write it, substrate utilization from the dx by dt type of equation. Dx by dt is the growth rate of microorganisms. Now, what I can do is I can also write down what is called suppose if I have, this is one equation what we have, this is equation A for the substrate utilization rate, utilization of substrate. Now I can also write similar thing like this. In a unit time, suppose if delta S is a substrate that is used, delta S is a substrate that is used in a unit time, okay, the rate of substrate utilization is proportional to the x delta T. Who is utilizing the substrate? Microorganisms are utilizing. This is a biomass concentration. Biomass concentration. Biomass is utilizing the substrate and hence there is a change in substrate. Change in substrate is because of the utilization of the substrate by biomass. So that is what proportional to x into dt, delta small change, elemental change. ds by dt, if I write it here, put limit delta t tending to 0, this will be equal to ds by dt is proportional to x, is proportional to x, or this proportionality I can change it by ds by dt is equal to a term called a q, I will put it as q into x. Now I define what is this q, q can be defined, so that means ds by dt, the food that is utilized is equal to q into x. The q is known as specific substrate utilization rate. Specific substrate utilization rate, okay. The units of this will be S and X are in the same units, that is what milligrams per liter, that is what we said, both of them are expressed in the same units. When we are expressing in the same units, then Q will take the unit of inverse of time. So this is inverse of time, time inverse. Q is nothing but ds by dt utilized over x. So this is what is Q value. Specific substrate utilization rate is this particular thing. Now we have an expression here. If I plot this graph, there is a similarity between this expression and another expression what is called a mu. Mu is the specific growth rate, is nothing but dx by dt, the growth of microorganisms, maybe I can write it here as growth, or g simply, I can write simply g, or growth I am writing it here, divided by x. So this is specific growth rate and this is specific substrate utilization rate, okay, I do not have to write it here, it is already there, okay. Now I would like to know what is the relationship between the specific substrate utilization rate and specific growth rate. Suppose, can you tell me what will be the relationship? The relationship can be easily derived. The specific substrate utilization rate and specific growth rate can easily be related. So how can we relate them? We can relate them using the yield coefficient rather. So the yield coefficient is given by y is equal to minus ds by dt, sorry, let me write it clearly, is dx by dt over x divided by ds by dt utilized. So I will divide both sides, both the numerator and denominator by x, that is what I am trying to do it, dx by dt over x, ds by dt utilized over x. So when I do so, what would happen? Y is equal to this dx by dt over x is nothing but dx by dt over x here in this particular equation, you can say that is equal to mu, and this particular thing ds by dt utilized over x is equal to q, so that is q. So that means this is the relationship between specific growth rate and specific substrate utilization rate and the yield coefficient. So in other words, yield coefficient is a ratio of, it is a ratio of specific growth rate divided by specific substrate utilization rate. Specific substrate utilization rate. That is what you can see. This is one relationship which is important. We will be using it quite often. So this particular relationship. Now let us take the equation, that is a specific growth, specific substrate utilization rate. So that is equation is equal to, that is what I am writing is Q is equal to dST utilized over X is the Q value. Okay. So that means, in other words, dST by dt utilized is equal to Q into X. And now this Q, specific substrate utilization rate, is not constant, is not constant. It is not constant, just like mu. Mu is not a constant, so Q is also not a constant. So the Q is equal to, Q is a function of substrate again. Q is a function of substrate. So this functionality is given by Q max S over Ks plus S. Q is equal to Q max S, Q max multiplied by S over Ks plus S. What is Q max? Q max is just like mu max. There is a similarity between the Q max and mu max. Q max is my maximum substrate utilization rate. This is a maximum substrate utilization rate is the Q max. Okay, it's similar to mu max, which we have discussed already. It is similar to that one. So there is a similarities between these two things. Now let us take that case 1 again, condition 1, when the substrate is unlimited, similar to the condition which we considered previously, which have got unlimited substrate. When you have unlimited substrate, what is going to happen to this equation is, again, that is S is greater than Ks and S plus Ks can be approximated to S as previously done, and hence substituting in this equation, maybe I call this equation as B. Substitute in the equation B for this Ks plus S, then this equation will be converted as Q is equal to Q max into S by S into X. So this particular thing is cancelled, this cancel out. So Q is equal to Q max times X, Q max times X. So that's what we have, that is Q is equal to Q max times, no, I am making a mistake, sorry. So I will make a mistake, I made a, this is Q max into, Q is equal to Q max into X, no mistake is done. So that is, this is equal to, this Q is equal to Q max, this X is not there, this is a mistake what I have made, please look at this. Q is equal to Q max S over S, that is in this particular equation I am substituting. So this Ks plus S is S, Q is equal to Q max. Q takes a value of Q max. So that means the specific growth rate, specific substrate utilization is a maximum value, it is a maximum value. So this particular thing I will substitute in the fundamental equation. The fundamental equation is the following, that is my fundamental equation is ds by dt utilized is equal to q into x. This is a fundamental equation. In this, now I substitute q max into x is equal to ds by dt utilized. Okay, this particular thing I will substitute in this particular q max into x. So, now this equation can be solved. Suppose if I plot the graph, that is equal to, or this particular thing, again I will take it this, if I take it to the bottom, q is equal to q max. This is one equation what way I am going to get. Yeah, this is the equation what we have, ds by dt utilized is equal to q max multiplied by x. In other words, if you say ds by dt utilized over x, this is nothing but for me q is equal to q max. Q is equal to q max. So, in this q is equal to q max, in this equation, this also I can write s to the power of 0, s to the power of 0, s to the power 0 order equation with respect to substrate. So, this is a 0 order equation with respect to substrate concentration. So, if I make a plot of this particular thing, I make a plot of this particular equation, it is something like this, that is I plot S here, substrate concentration, and I plot Q here. Q is nothing but ds by dt utilized divided by x, specific substrate utilization rate. This is ds by dt utilized over x. So, if I take this, when S is unlimited, when the substrate concentration is very high, that is what we have taken a case, when the substrate concentration is very high, case number 1, then this is independent of substrate concentration. This is 0 order equation. This is a line like this will come when the substrate concentration is very high. So, this is my q is equal to q max. Q is equal to q max, and this is the 0 order with respect to substrate concentration, 0 order with respect to substrate concentration. That is the curve what we get it here. So, now this is one condition always, that is availability of food is plenty of food is available. Now, when the food becomes limited, that is case number 2, now I will take it here, case number 2, case number 2, when S is limited food is available. And again I would like to reiterate the fact that most of the times in domestic wastewater treatment we come across this particular thing, when the food is limited. When the food is limited, now we can say that equation whatever we have, ds by dt utilized is equal to q max S over Ks plus S multiplied by x. In this particular equation, what I will do is S concentration of S is less than Ks. When it is less than Ks, I can say Ks plus S is equal to Ks itself. So that this equation becomes now ds by dt utilization of substrate is equal to q max over Ks, q max over Ks, Ks plus S is also Ks. So, q max Ks multiplied by x into S into x, that is the equation it will become. So, q max is a constant, as we know, Ks is a constant, the whole thing is a constant, q max by Ks is a constant, this particular bracketed thing is a constant. So, this constant may be called as a k, I can call it as a k. So, now again taking that equation, we have ds by dt utilized divided by x then immediately this becomes Q, that is a specific substrate utilization, that will be equal to now K times, that is a K, Q max over Ks is K, that is what Q max over Ks is equal to K multiplied by S, multiplied by S. So please look at this equation and in the previous equation, this is one equation which you have to look at. So this equation is a first order equation with respect to substrate concentration. It is the first order with respect to substrate concentration. So if I plot a graph, this Q that is in this plot I will go S versus Q, if I plot it, this is a first order equation, this okay, this is the first order equation, that is Q is equal to Q max, no, Q is equal to not Q max, now I have to write it as K max, or I can write it as Q max over Ks, Q max over Ks into S. So this is S to the power of 1. So this is a first order equation, first order. Now when the substrate is limited, it depends on the concentration of the substrate, that is substrate utilization depends on the concentration of substrate. When the substrate is unlimited, the utilization of substrate is independent of the concentration of substrate. This is a 0 order, this is a first order. So in between, this is a first order should become a 0 order, or 0 order become first order. This particular thing is a discontinuous, that means there is a some other order here between these two things, connecting these two things, and hence this model is known as a discontinuous model for substrate utilization. For substrate utilization. So why it is called discontinuous model? It is 0 order when the substrate is plentifully available, and it is a first order model when the substrate is limited. In between the situations, it is a discontinuous model. So that's what it is said. So that means we have got two equations now. One is the specific growth rate, another is the specific substrate utilization rate, and two cases are considered in both cases. One is when the substrate is limited, when the substrate is unlimited, and these two things are connected by what is called a yield coefficient. Yield coefficient is very, very important, and we have seen that particular thing. Now before one more point I would like to discuss before going further. Suppose if you give a substrate to the microorganism, I give a substrate to the microorganism, the microorganisms could be aerobic or anaerobic, depending upon that the value of Y will be different. So let us take a part of this is used for energy, another part is used for the biosynthesis. That is what we have been saying. Okay. If you see further, this amount of energy that is produced is used for two purposes. Okay. One is what is called energy required for growth, of microorganism. Growth of microorganism increases the biomass weight of the biomass. Energy required for the growth of microbe, that is one thing, and second thing is energy is also required not only for growing, energy is also required for maintenance. So energy required for maintenance. So maintenance means the cell has to do certain amount of work. It has to move from one place to another place, okay, using the flagellum, okay, it has to do certain other works. So this is called energy required for the maintenance. So when this energy is called maintenance energy, this energy is called growth energy, energy required for growth. GE is the growth energy, ME is the maintenance energy. When I give a substrate to the microorganism, the microorganism utilizes the a part of the substrate for energy, that energy goes for the growth of microorganism as well as growth of maintenance energy. Now the situation is the following: if I do not give any substrate to the microorganism, if I give the substrate, they will utilize in this fashion. Now what would happen is that there is a condition already which I have shown, that when the substrate is completely exhausted, when the S is completely exhausted, when there is no food available for the microorganisms, what do the microorganisms do? Okay, definitely you can say that the microorganisms do not grow in number, large size, they will not grow, no growth takes place, growth ceases, cessation of growth disappears. However, there is necessity for the maintenance energy, okay, maintenance energy is to be met even if there is no substrate, because the cell has to do certain amount of work, whether it is it is not growing in size because there is no food, but it has to be alive. For alive it has to do certain work, so that maintenance energy is required. This maintenance energy is required for what is called a basal metabolism. To maintain basal metabolism is the minimum metabolism that is required for the cells. So what I will do in the next class is that we would see how this maintenance energy is met, okay, when the substrate is not available for the microorganisms.